Hello, Assalamu alaikum. In this video, we will understand about the symbiotic nitrogen fixation. So, how the symbiotic bacteria or other organisms will interact with the chlorophyta, the photosynthetic plant. The function of nitrogen fixing plant and microbe association we will discuss in this video, and endosymbiont and ectosymbionts if you know about that. But we will not understand about the endosymbiont and ectosymbionts, but we will understand about the function of nitrogen fixing plant microbe association. So this is the symbiotic nitrogen fixer is the endosymbionts. This endosymbiont is the alpha proteobacteria and beta proteobacteria and the actinobacteria, and the cyanobacteria is the frankia also. The alpha proteobacteria is used for that plant is known as the parasponia. The parasponia is the host, while this is the photosynthetic organism, consume the carbon dioxide and produce the oxygen, while the sugar is the basic product and nutrient exchange within the symbiotic mutual relationship for the nitrogen fixer. Anyhow, this is the actinobacteria used for the actinorhizal plant, legumes, and cyanobacteria used for cycas, the basal eudicots, and bryophyta. The plant growth promoting rhizobilaria is the PGPR. Is associative or rhizospheric colonizers, azospirillum and nostoc. This is associative relationship or associative or rhizospheric colonizers and azospirillum nostoc are, and the endophytic mean the inside of the plant is the colonizers azoarchus and the gluconectrobacteria and the herbaspirillum and nostoc, which that is used for the cereal crops is the zea, oryza sativum, zea maize, saccharum is the sugarcane and ferns, azolla, bryophytes also. This is also the photosynthetic bacteria which that will give the sugar to the symbiotic nitrogen fixer, while the nitrogen fixer provides the ammonium ion through nitrogen gas to fix it. Nitrogen dioxide and water also used to fix it. In this way, they also provide the water. This is a plant contain shoot system and root system. Root system contain nodules. Nitrogen fixer bacteria live in the nodule. So the development of the nodule is very important. Is known as the nodulation. This nodulation occur with the passage of time due to the growth of bacteria as well as growth of the plant. This is the bacterial cell. Let's understand about the nitrogen fixation and nodulation growth. So in this video we will discuss about the nitrogen fixing and nodulation, symbiotic nitrogen fixation, adaptation. This nitrogen atmospheric will move through a nitrogen transporter into the bacterial cell, and the nitrogenase enzyme will convert the nitrogen into the ammonia, and in this way the oxygen will inhibit it, while the atmospheric nitrogen fixation will occur will lead to, here is the DNA contain nitrogen nitrifying fixation cytochrome and BD gene. Anyway, this is a very important functional process. This is the phosphatase transporter, the phosphate transporter. This ammonia will convert due to the glutamine synthase, glutamate synthetase enzyme to produce amino acid with the fixation of carbon monoxide also. Anyhow, this is the Krebs cycle can be occur in some bacteria will produce ATP, carbon dioxide, but the ATP is used for the nitrogen fixation, and in this way the malate is used from the plant oxaloacetate as well. And phosphate also provided via a plant, but the phosphate also present in the soil. This ammonia will move out, will convert into the ammonium ion, by the accepting of the one proton more. This can be moved again inside while the ammonia will move into the plant cell, into the root. Anyhow, this basically here is the glutamate conversion from the ammonium. Sorry, glutamate is used also for the Krebs cycle to produce the asparagine, and this asparagine also, and in this the glutamate also glutamine. Sorry, this glutamine synthase enzyme used to glutamine. In this way, the normal compounds will form and the plant will be grow. And in this way, remember, this is the synthesis of protein due to the nitrogen fixation. Anyhow, this is the sucrose due to the photosynthesis convert into the hexose and phosphoenol pyruvate and oxaloacetate. Carbon dioxide fixed to produce malate and DCTA transporter will malate accept into the Krebs cycle in the bacteria. Anyhow, phosphate also transport within the bacteria and plant. This nodulation A, B, C, and a node gene and a node L and nitric oxide and other genes basically are present in the bacteria, while here is the symbiotic gene will produce cytokines and nodule organogenesis pathway and biosynthesis of flavonoid also produced from the symbiotic gene of the plant will move in toward the bacterial gene activation. The bacterial node A, B, C, and the node L, N, O, E, and D, which that is also important for the activation due to the flavonoid production from the biosynthesis of flavonoid from the symbiotic gene will activate to produce a node proteins, which that is important for the nodulation. So the nodule formation when the node factor will bind with the receptor will lead to nodule organogenesis. In this way, the symbiotic nitrogen fixation is very important for the protein containing beans and legume plant. And in this way, we have discussed about the symbiotic relationship of nitrogen fixation and provide the amino group to the plant, while the plant will provide the sugar as energy in the form of malate and the glutamate. Thanks for watching. See you in the next video. Bye.